I will try to be brief, but also I will try to give some messages. Indeed, all the presenters in the previous, or all the speakers in the previous lecture have defined what we call two different pillars: public health, community nutrition, versus personalized nutrition, personalized medicine, etc. Now I am going to talk more in the second area. What happens when we are talking or we are thinking in personalized nutrition? For that reason, the topic that I am going to develop in the next few minutes will be the Nutrigenome and obesity. The first question, when we see this picture, this is from a famous Colombian painter, we can think that this family, this is the title of this painting, is obese because they eat a lot. Probably because they are sedentary. Probably, and it's easy to think, that they are obese because the family has some genetic predisposition to be obese. But this is a very nice picture. Some of you know, because it's not the first time that I talk about that. What happened with the pet? Then we can have interaction between genetics, but also with lifestyle predisposing factors. In other words, genetic is important, but it should be placed in their own side. Therefore, I am going to talk about these interactions. And in the next few minutes, I am talking that really the nutrient balance depends on many different factors. One of them is genetics that affect nutrient intake, but also nutrient requirements. In other words, the homeostatic balance in our body, in our organism, depends on genetic equilibrium. Given further, we know that nutrients provide energy, provide structural materials, but also provide regulatory elements. Indeed, some of these regulatory elements, like fatty acid, retinoids, vitamin D, glucose, energy, iron, selenium, minerals, vitamin in general, affect transcription processes and also translation processes or even post-translational mechanism. Genetics is influencing gene expression. And different experiment has been able to demonstrate that through a specific receptor, either in the membrane or in the nucleus, through intermediate metabolism or through transcription factors, nutrient can influence gene expression. There is no doubt. Nutrition have an impact on genetic responses. To make clear, this phenotype, obese or disease, against healthy or lean, this phenotype depends not only in the genotype, on the nucleotide sequence, but also depends on environmental factors, environmental interactions. Then every body, every person, every organism depends on their own genetic makeup, but also the impact of external factors. To make more clear this idea, we know now that dietary choices and food intake depends on genetics. We know that metabolic process depends on nutrients, but also nutrition influence aspects like digestion, absorption, transport, etc. that depend on genes. In other words, there are possible interaction of dietary intake with genetic variability to affect disease risk. What has happened in the last 10-15 years? That the investigators have been able to make the sequencing of the human genome. Subsequent analysis has been able to demonstrate human genetic variations. Some studies have identified that gene variants are associated with disease markers, and also we know the impact of nutrition on gene expression. In other words, genetic makeup can determine unique nutritional requirements and response to different foods. But this idyllic picture is now complicated by what we call nutri-epigenomics. What is epigenetics? What is nutri-epigenomics? Indeed, epigenetic processes modulate gene activity and expression without important changes in DNA nucleotide sequence. That is very, very important. The sequence is important, but not only. For that reason, I have prepared a film in which the English and the message could be better than the message that I am giving to you now. Sorry?
Epigenetics refers to the heritable modifications in gene function or activity without changes in the DNA sequence. Epigenetic processes are genetic modifications that affect gene regulation by changing the DNA conformation. Epigenetic changes are found in many different cancer cell types. One such change is histone modification that may be caused by overexpression or aberrant recruitment of HDACs. Within the chromosome, DNA is packaged into chromatin. Chromatin consists of DNA, structural histone proteins, and non-histone proteins. Within chromatin, the repeating unit is the nucleosome. Nucleosomes are made up of 146 base pairs of 2 superhelical turns of DNA wrapped around a core of 8 histones. The histones are responsible for maintaining the chromatin's shape and structure. Epigenetic modifications, such as histone acetylation, occur at the amino-terminal tails of the histones that protrude from the nucleosomes. Acetylation of histones is generally acknowledged to play a key role in the regulation of gene expression. Histone acetylation is controlled by the balance in the activity of 2 enzymes, histone acetyltransferase, or HAT, and histone deacetylase, or HDAC. For a gene to be transcribed, it must become physically accessible to transcriptional machinery. Acetylation of histones by HAT causes uncoiling of DNA and an open chromatin structure. This causes genes to become accessible to transcription factors, allowing gene expression to occur and proteins to be made. Conversely, deacetylation of histones by HDAC results in tight coiling of the DNA and a closed chromatin structure. In some cancer cells, there is an overexpression of HDACs, an aberrant recruitment of HDACs, or an underexpression of HATs, resulting in hypoacetylation of histones and therefore a condensed or closed chromatin structure.
Thank you very much. Then, continuing with the same idea, but in the Spanish way, with my accent, we are talking that DNA methylation and histone modification can affect gene expression. In other words, how the genes are read. But not only DNA methylation and histone modification, but also chromatin packaging alteration and situation around the nucleosomes can affect the gene expression. You see, we have the same nucleotides, but some of them they are methylated, some of them they are acetylated, or they have other covalent modification. In other words, in addition to the genetic code, we have the histone code that depends on how the chromosome or the chromatin is packaged, but also depends on the presence of methylation, phosphorylation, acetylation, etc. What is clear is that this histone modification involve different enzymes and donors, some of which depends of nutrition. Another important complementary idea. You know that nucleotides belong to us all our lifespan, during all our life, but changes, epigenetic marks, are not permanent. They can be changed by maternal care, by aging, by dietary compounds, by toxin, by inflammation. All of them regulate methylation. And there are specific windows in our lifespan in which the situation of DNA methylation or erasure of methylation can appear. For all of you, the message is that epigenetic changes are not permanent and can affect to different genes. There are genes that can be methylated, some of them cannot be methylated. I am going to show you very simply and rapidly an experiment carried out at the University of Navarra a few years ago. We fed rats with a controlled diet and with a cafeteria diet, rich in fat, in sugars, etc. And of course, the animals were obese. After that, we have taken DNA, we have measured methylation status, and what is clear is that those animals receiving the cafeteria diet, the high-fat diet, have increased level of methylation in this specific gene, leptin. And what is more clear is that doing more investigation, we have the following message. High-fat diet induced obesity in rodent is able to enhance the methylation pattern of the leptin promoter in adipocyte. Very important. And second, those subjects more methylated in the high-fat diet show lower leptin levels and lower body weight. In other words, methylation can influence the phenotype affecting leptin. This is important. A diet rich in fat can provide not only energy, can make the animal fat, but also can affect the future regulation. As you see, there are many dietary factors, drugs, metabolism that could affect not only methylation but also histone acetylation. And also here we see that maternal alteration, methylation, microminerals, a vegetarian diet, fatty acids can influence different aspects of epigenetics, either methylation, phosphorylation, acetylation, etc. Now a few examples, quick few examples. For instance, supplementation of a maternal diet with genistein, this is a flavonoid, produce alteration in DNA methylation that make that the color code are different. You see, yellow and dark. Depends on the methylation. Or this is an example that feeding animals with a cocktail containing folic acid, vitamin B12, betaine and choline induce DNA hypermethylation. But what happened is that after 3 generations, those individuals supplemented have less possibilities to become obese than those subjects that are unsupplemented. A mixture of promethylating agents affect the body weight of the progeny. Another example. Maternal care, licking, grooming, nursing methods. I mean, the number of times that the mother put the tongue into the pups may affect methylation status of an important gene like glucocorticoid receptor in the hippocampus. Maternal care. Also, we see here that methylation changes with age. This is done with twins, and when they increase age, they increase the percentage methylation. All of this, I am saying all of this in order just to keep one unique message. Methylation changes with age, with diet, with perinatal condition. But also, this information about epigenetics could be useful for medical purpose. You see, these two human beings wish to be leaner, to eat more during holidays or the Thanksgiving Day or in Christmas. This is not the same for the duck, that he wants to be lean for other reasons. We select again one gene, the TNF-alpha promoter. We fed different individuals, and which were the information that we get? Subject less methylated lost more weight. Can be as a prognostic. Subject with more baseline TNF-alpha have the promoter more methylated, less capacity of control. Subject with the promoter more methylated decrease less the TNF-alpha levels, less capacity of control. This has been used that before and after the diet, there are some genes, these 5 genes, that show different response depending on the diet. Methylation changes with the diet. But also, the epigenetic biomarkers can be used to detect low versus high responder, to identify people that respond differently to a slimming diet, to a energy restricted diet. You see, in blue, subjects that are leaner and those that they don't respond to the diet. The methylation of some genes are different. No, because the methylation. To summarize, I promise that I am going to be brief, and I will be brief. What I am trying to say to all of you is that the situation is changing. we think that many diseases depend of nutrition, depend of sedentary lifestyle, but now, to complete this picture, we need to consider genetics, SNPs, polymorphism, genomics, gene expression, but also epigenetics. Again, personalized nutrition, therefore, need to take into account genetic background, of course, family history, of course, likes and personal dislikes, previous disease, the history of everyone, culture, allergies, intolerance, physical activity, but now epigenetic marks appear as a new marker. I am summarizing and I try to give you the last smiling in your face. Of course, we try to demonstrate that a diet with a variety of foods is important. Of course, to maintain health, it's important to make exercise, not watching program about sport, eating a lot of food. You see, this looks good. It's a 6 hour special on how society is becoming too sedentary. Watching Olympics is good, but that no help too much. And of course, genetics is important. But you see, this physician is saying, frankly, I am a bit confused. According to the genetic printout, this gentleman is in fact a goat. Take care. The information is there, but they need to be carefully interpreted. Of course, when you don't know what is happening to anybody, please don't say that it's due to epigenetics. Have some influence, but it's not all. Okay, thank you very much.